we'll first actually go through very briefly the curation process that we have agree on in the consortium, just to remind people a little bit about that. And then I will talk about the data model relevant to the sequencing data, and then use some sort of like simple examples to try to give some context in terms of how the metadata is supposed to be filled in, and then we'll do a walkthrough through the whole submission process. This is actually it's going to be a summary of the curation process that we have an agreement with among the PIs and the consortium. So the vertical axis is the stakeholders involved in the data submission process, and the progress proceed from left to right. So while you're preparing your data, you can mark your records as in preparation. So then that way, we know that it's actually a draft state, and nobody can actually look into it, that in detail. And then once that is done, depending on the lab, agreement, you can either mark them as PI review so that the PI can actually take a final look before changing the status to submit it. Or if the PI actually grants you the authority to just go ahead and submit the data, you can then mark them as submitted. Once it's submitted, then it's basically turned into our, the hub and BioCurator to look at the data. So depending on the different data types, sometimes the data will automatically get released. These are basically usually are things that are kind of like files, files properties, things that actually have very descriptive information and doesn't have a lot of biology metadata. For RNA-seq, things, records that has a lot of metadata, biology metadata, such as experiment or antibodies, then it will actually go to biocuration review. And in that case, Todd's going to take a look at it. If he finds something that's actually missing, he will mark them as amendment and then contact the lab, to have the data corrected. Otherwise, he will release the data. Until the data is released, nobody can actually, only the consortium member can see the records. Once it's released, then the public can see them. So, so that's pretty much kind of the summary of the curation process. And you will see the field curation status in almost every records that you'll be entering. Okay, so this slide summarizes the data model relevant to the sequencing data. The blue one are the core of sequencing data models, and then the yellow boxes are kind of more generic or other high-level data model entities. So in the sequencing data, we use different data types to capture the process of capture metadata of the sequencing data. At the high level, we have a study that this is equivalent to GEO series. If you're familiar with GEO, so it basically describes the goal of the study. It describes the overall design. So that's basically study. And then within a study, you can have multiple experiments associated with it. The goal of experiment is to kind of capture the micro experiment done to a set of replicates. And usually these set of replicates will have exactly the same protocols, same setting that actually are done to that, right? And then an experiment can have one or more replicates or a single cell matrix index for the single cell sequencing path. And then for the bulk sequencing, for example, mRNA-seq, ChIP-seq, or ATAC-seq, so you can see that actually this path, right? So you'll be creating replicates and then attach them to the experiment, and then all the sequencing files are then attached to the specific replicate that they actually are used for the sequencing, right? Similarly, for the single cell path, it's the same concept. So you will have all the files or the sequencing files and analysis files that actually attach to specific cell metrics, and which also is hooked up to the experiment, right? So those are kind of the main entities that we'll be dealing with. On the right-hand side, those are kind of metadata associated with the either study or the experiment. So so that was a question that come up during the data submission process with prior data submitters. So basically, they were asking a question of, if you have the analysis file that are kind of summarize the whole study, not specific to a single cell. So what should we do in that case? So we created analysis files at the study level for you to submit arbitrarily analysis files or any other data summary files that you want to share with other people. And then for these files, you can actually hook it up to the study. And then associated with experiment, there will be experiment settings that actually capture all the information about alignment, library, quantification, sequencing methods, and visualization. Okay? And then for some type of sequencing data, you might want to provide antibody information relevant to the cell isolation. So this is the place for doing that. And then custom metadata is also provided if you want to actually just, capture your own customized metadata that you want to use internally for your own lab. And then another important thing is that the replicate and single cell metrics will be include information about this sample, the viral sample, and that actually through the specimen data model, which I'm going to briefly summarize next. So far, is there any question relevant to this high-level metadata model?
Yeah, this is Michelle. I have a question on the study level analysis files. Like previously when we submitted things, it was a field, a drop-down field, and you could type in a descriptor of what the overall experimental plan is. Is that what you're talking about there?
No, so basically the study will have a field that actually is called overall design, and I'm going to show you, like a detailed metadata associated with the study later. The analysis files are the actual files that you can upload. Like, for example, if you have Excel that actually, summarize some of the analytics, or you have like clustering. So in the single cell cases, if you have like clustering information that you want to kind of attach at the metadata at the study level.
And you're saying that that's mandatory, because in the past we have always talked about analysis files not being submitted because there's so many different ways you can run the analysis.
No, so this is not mandatory. This is really just a place for people that actually want to share. You can actually hook it up to the existing set of data.
Okay, I thought by your blue text, but it's only the blue text with the asterisk that's required.
No. Okay. So the mandatory means the stars. So I'm sorry. So it's really mean that, and I'm going to go over that in the subsequent slides as well. So according to our agreement, so all the raw sequencing files and BAM files are required.
Right.
Everything else, if you have, they are encouraged to be submitted.
Okay.
Be shared, but they are not mandatory.
Okay.
Right. So then these are kind of like raw analysis file relevant to a specific sample, right, or a specific replicate.
Okay.
Some questions came up that if you really aggregate across all these different replicates or experiments, so where do I put that data analysis files? So this is actually the place that they will be putting up those files at that level.
Okay. All right. Thank you.
Okay. So then, so this is actually summarized the specimen data model. We will have the next tutorial that's specifically focused on specimen and imaging data. So I won't go into detail, but just to point out that specimen is actually used to capture all the biosamples in the consortium. So all the imaging data will be linked to specimen, legacy microarray data, and legacy RNA-seq are linked to specimen, and also the current model also linked to specimen. And then on the right-hand side, again, there are like metadata that actually are relevant to that specific specimen. So the one the one that we care most about in the sequencing data context are the mouse strain and the allele that actually is used to capture the genotype. Okay, so that is also very important. Without this, then we wouldn't know the stage or the species of the sample. So now I'm going to talk about some of the constraints in the model and some of the simple examples, just so that hopefully will give you a better idea of how the model is used. So for the bulk sequencing data, like for example mRNA-seq, we assume that a study can have one or more experiments. We'll have one or more experiments, and then an experiment can have one or more replicates. And then we actually capture both biological and technical replicates in the replicate metadata. And then all replicates within the same experiment share the same protocol and experiment settings. And then files are attached to the associate replicates. Okay, so in this example, let's say you want to do sequencing data for injury model of mouse kidney E16.5. So assuming that we will be doing this for two groups of samples, one for the control and one with the injury model being applied to the samples. And so assuming that it's going to be done to like a set of replicates, right? And so in this case, then we will have three different replicates. All of them have one technical replicate, but the biological replicates will be changed from 1, 2, and 3. And then each one of the replicate then will point to a different specimen. Okay, and then we'll be doing the same thing as the injury model one. So in this case, when you go look at the data, you will see a total of 6 replicates associated with the study. Okay, any other questions? Similarly, for the single-cell RNA-seq, study can have one or more experiments, and then an experiment can have one or more single-cell metrics, right? And then the metrics is similar to replicate. It has biological and technical replicate numbers, and then all single-cell metrics within the same experiment share the same protocol and experiment settings. And there's a note field that you can put some more description on how the metrics are derived. And then the files are attached to the single-cell metrics. So I'm providing 2 examples, and those are actual examples that we have in the repository. The first one is from Andy's lab, and it is basically sort of supporting mouse samples in the study of cellular diversity in human nephrogenesis. So in this case, he has 4 different experiments, and each one of the experiment only has one single-cell metric, and it's hooked up to different mouse samples. So that's one example. And then another example is from Ben's lab. So study single-cell RNA-seq of ABMR biopsy. So in this case, he has only one experiment that actually is being done to 4 different technical replicates. Okay, so I think the model captured both cases. Any questions? So far so good. So then the procedure for submitting the sequencing data. The first step is to basically make sure that you join the kidney writer group, and we do have detailed instructions in terms of how to follow all these steps on our website, provided at the lower right corner here. So first, join the kidney writer group if you haven't already. The PIs will have to basically let us know whether to approve the lab members, and then once you're actually in the writer group, then you can create data and upload your data. The second step is before you can upload the data, you have to create metadata records for your sequencing data files. So these include study, experiment, and all the green boxes that I mentioned. And then once all the metadata records are created, then there are 2 ways to upload the files. One of them is to use the usual browser GUI that you use to create the metadata to upload the actual files. And the other one is to use the client tool that you'll be installed on your computers, and then to upload the files to the server. So we're going to go over all of this in detail later. So supporting 5 formats. So pretty much, I think we support everything that you want to upload, right? But I think the primary thing that kind of been recently discussed are kind of summarized in this table. So we want the raw sequencing file, FASTQ files. R1 is used for FReads, and R2.FASTQ.gz is used for RReads. And then the .bam is the alignment files, and the rest of them can also be submitted. There will be a file type field if you actually submit that manually through the GUI, or the file extension is used to extract the file type from your data if you use the client tool. Okay, so now I'm going to do a walkthrough through the creation process. So the first thing that you need to do, you need to remember, is basically the landing page of the consortium. So either goodmap.org or rebuildingakidney.org will lead you to the same data portal. So clicking on any link under resources on either site, or clicking directly to the sequencing data will lead you to the data portal. And then common among all these different data types is the navigation bar, right, that allow you to access all these different data types that we have in the repository. So to get to the study, existing study in the system, you would actually click on search, and then click on gene expression data, and then it will have options for sequencing data, and then choose a study. It will lead you to the study page. It look like this. The one on the left panel allows you to actually select the search criteria, and then the right panels show the result of the existing study that match that criteria. So Chris made some video tutorial or has a wiki page that kind of describe how to use this feature in detail. So I won't go over that, but just to summarize that we have the search page for that here. So and then if you click on any of these it will lead you to individual study. For example, you can see that this is just an example of existing study. It's actually has an internal ID, and then it has a title and summary, and the overall design is actually it's also here as well. But and I will show you the complete form in the next slide. But basically, the metadata that we use here, we extract that from the geo model, just to make sure that our final goal is to be able for you to export your study so that the final product will generate an Excel files that is required for geo submission. We're not there yet, but that's but at least the metadata here is provided so that kind of information can be submitted to our system. Okay, and then we have the record status and curation status, like I mentioned. The record status will have complete or incomplete, and if it is incomplete, it will provide more information in terms of what is missing. And then the right panel is more like a table content, so you can see all the relevant records related to the study. So in this case, you can see all the experiments actually that's been submitted to this study, and then the study analysis file. In this case, there's nothing. So to create a study, right, there are multiple ways to do that. So first, the easiest one is basically to go to the create menu and then choose the sequencing file, sequencing data, and then study. So it will lead you to an empty form that has the columns, has the fields, and then you're just supposed to fill in the blank, right? Asterisk actually indicates that those are required, and until you provide that information, it won't allow you to submit. And if you want more two tips, you can this dotted line you can see here, it indicated there's a tooltip associated with that particular field, so provide you more information about what it's intended to capture. Another way to create a new study, for example, is sometimes you don't want to keep entering similar information many times. You can use a copy. So this will allow you to sort of create a new one based on the existing content of an existing record. So so what I'm showing here is basically a result of clicking copy of this record, and then it basically just pretty much pre-fill everything using the original one, and then you can change whatever that is different. So once you actually click submit data, then it will lead you to this page, and then except that the experiment will be 0, right? To add a new experiment, just click add on top of the relevant table, and when you do that, it will basically pop up a form with the study already pre-filled. So that way you don't have to go in and try to find which study you're talking about, which is automatically fill that in. And then again, just fill in the rest of the details. So in this case, it asks for another experiment internal ID, the name. This is more like human readable names that actually it can use. Some of the old data is we use the internal ID because the name was added later, based on the legacy RNA-seq data from GUDMAP. And then it asks for the sequencing type, species, and things like that, as well as all the protocols, right? If you use any published protocol as part of your experiment, you can then link to that by creating a treatment protocol reference. And when you click on that, it will basically prompt you to choose from existing publication or create a new kind of citation reference. You can also just write, like a short summary of the specific protocol that's used for the experiment as well. So that's pretty much the main content of the experiment. And then once you click submit, then it basically just fill in the main content of the form, and then all these related tables will be empty, right? So I'm just showing a result of an existing experiment here, just so that it has a little more detail. So then you can see that relevant to the experiment, there's experiment antibodies, custom metadata, experiment setting, replicate single cell file, and scRNA file, right? Click on any of these things will lead you directly to that table. So in this particular case, if you actually click add on any of this, on top of this table, it will directly lead you to add new entries and then add it to that table. So I won't be going into detail about experiment setting, but pretty much if you click on it, it will basically give you a form that asks about the alignment, format, aligner, genome, reference genome, and such, and then also library sequencing methods, like that. I will focus more on the creating new replicates, but it's exactly the same process for any of these records. Okay, so clicking on add replicate, it will lead you to this form. Okay? So again, it asks for the experiment ID is automatically filled in. It asks for the replicate's number and some sort of summary of the replicates here. The most important thing is basically the specimen ID, the specimen. So in this case, you would actually, if you click on the any of this highlighted by this red box here, it basically will lead you to a model, a pop-up window that show an existing specimen in the system. You can either choose from existing specimen, but most likely your specimen will not be there. So then what you would do is basically then add it to the specimen, right? Try to search first. If it's not there, then you actually add it to the collection. So then click plus here. It will lead you to another page to create a specimen. And then after the specimen is finished, then this list will be updated, and then you can then choose it to add to the replicate. That makes sense? So this is just to quickly show the specimen form. So similarly, when you create a new one, it has the core metadata relevant to the specimen, for example, the species, the stage, assay type, things like that. And then once you click submit, then similarly, you see this kind of record, the detail, right? And then in this case, we add the specimen allele and mouse strain by clicking add on top of the relevant table. And I think these are the two main things that are needed for the sequencing data. So, so there are also most likely more than one replicate will be created. Sort of facilitate this bulk creation. We do have some nice feature that when you first creating, there will be only one form showed up, and as you actually click on the plus button, it basically replicate the last column that you see on the form. So then that way you can actually add 3 replicates simultaneously, right? So then and then just change whatever that's different for this different replicate, and then click submit. Then the 3 of them will be created at the same time. So I just find that very useful feature. Same thing for single cell metrics, a slightly different form, right? So apart from the replicates number and specimen, it's also asked for some like basic analytics like map reads, reads per cell and stuff like that. And there's a note field in here that you can use to sort of provide further information in terms of how the analytics, how is the data derived from the sequencing data that you have associated with a single cell metric. and that's the detail of the single cell. So it has a little bit more fields in here. So that is pretty much like the core things that you need to do in prior to uploading the sequencing files. Any questions?
No questions about this. So I guess the only thing is this presentation by PowerPoint will be distributed to everyone, right?
Yeah.
Okay.
So the next section is going to be talking more on how to upload actual files. And then, like I mentioned, there are two ways to do that. I'm going to be showing the one actually done through the GUI, to the browser GUI, and then Laura's going to take over the 3.2, which is to use the client tools to upload bulk, like huge sequencing files. So, like any other records that I've shown so far, right? So you see files associated with the replicate, and then when you click on that, it will actually pop up a form, right? And then you can do one or multiple files, right? And then you will see in the form a select file, a file menu that provide ability for you to choose a file in your system and then upload it through the browser. So this one is convenient, but at the same time, this is actually done through the browser, so there's some sort of limitation on the speed. So it could take a long time. I've done some sort of experiment, and if you have 5 GB of files, or 5 to 10, I think it's doable. But if your sequencing files are like 50 GB, that could just take too long, so that might not be applicable. But this is one way to actually upload the files or to modify the files. So and until we actually implement some sort of retry logic in here. So until you close the tab, if the submission failed midway, you can actually click submit again, and it basically try to pick up from where it was before. Another nice feature that I just like to introduce to people is that usually along with the ad, right, on top of this sort of related entities, you see view more. When you click on that, it actually will lead you to that specific set of records of that type, right? Pretty much it pre-filter the search criteria so that you can get to the set quickly. So in this case, when you click view more, it will get you to the files relevant to that replicate. And then notice there's an edit button on top of the search page. If you click on that, it will lead you to a form that actually has the content of existing data. And then you can actually make modification. For example, if you want to change curation status, you can do all of that and then click submit to change to modify multiple records at the same time. So that's kind of the way to upload the data from using the GUI. If there's no other question, then Laura can take over for the second way of uploading the sequencing files. Okay, Laura, you want me to stop screening, sharing, I guess?
Okay. So I'm sharing my screen now, and I just want to verify that you can see a Chrome window and another big yellow window. Can you guys see all that?
Yeah, but we cannot read them. They're too small.
So I can't really hear you that well when you talk. My audio is really bad.
Yes, we can see them, but everything is so small we can't read it.
I think you see both screens of yours.
Do
you have two monitors?
Yeah.
That's weird because I've made this one in particular really big. Is it that it's taking up a small amount of the screen space, or is it that the text size is small?
Looks like you have two monitors, each with a different background, and your Blue Jeans window is on the other monitor from your Chrome and your yellow screen.
Yeah, unfortunately, I don't know how to share only.
Maybe move them all on the same monitor.
I guess what I can do is
way to be share, Laura, then it will be one monitor only.
And so how did I really can't hear you when you
Set the
talk
display.
really loud?
Set the display so that it's only, it's mirror each other. So there's only one monitor active.
Yeah, that's an option. Let me unshare for a minute. You're sure you don't have any questions for what we've been going over so far?
Well, here's a real basic question for Hangsuda. One of the data types you showed was analysis files, sort of the final results.
Right.
So for me, that's like an Excel spreadsheet with the fold change and the p-values and all that for all the different genes. Is there a specific place I put that? I think those files are actually the most usable to an end user, and maybe people want to validate them by going back to the primary data, but those types of spreadsheets are sort of immediately usable. Where do we put them?
So basically, if those are specific to the each replicate or the single cell replicate, then you can just hook it up there. So then it will be showed up along with the other sequencing data. But you can also submit like the study-wide level, at the study analysis files. So that's really just kind of like a dumping ground for arbitrary files that you can just write the description and then have them shown when you actually go to the study.
So is there a specific address where we upload those to, or button to push?
So uploading the files, it's the same. So it's the same thing as actually going. If you want to upload it to the study analysis file, then you would just be going to the under the study, going to the add on top of the study analysis file. When you click on that, it will basically prompt you with a form, right? And then you fill in the form, and then there'll be a file selector. That's why you upload the file.
Okay, great, thanks.
Okay, and Laura's going to show you a different way of uploading through the client tool as well.
Okay, can everyone see better now?
Yeah.
Okay, and just this is the window that when Hangsuda was talking about. So you would click the add here to add a
It's a little bit better, but you need to make that window as big as possible because right now it's still
okay. We're
pretty
actually
hard
not
to read.
That window. So actually, what I'm about to show you isn't going to actually use these windows. What's important is can you read the text in this window, this yellow window?
Pretty much.
Okay. I just want to show that we have a wiki page that has step-by-step instructions with screenshots for everything that I'm about to tell you, and that is linked from the slides that will be sent out today. So just so you know, this is actually online. So what we have is two ways of using client tools to upload the files. And again, as Hangsuda said, if you want to, you can actually just go through the graphical interface, the web interface, and upload these files the way that you would upload any kind of image files. But if you have a lot of files, or if you have big files, or if you just want to organize them on your local systems before you upload them, you can use these client tools. And there are two flavors, basically, of the client tool. So one is to be used if you have all of the files that you want to upload on your desktop system. And if you do that, there's a tool that you can download that will give you a nice graphical interface that you can use to interactively select the database or select the folder where the files are and upload them from there. You get nice, pretty screens and help. The other option is if all of your files are on a remote server and not on your desktop, so you can't access them through a desktop interface. In that case, we have a command line tool that you can run on that remote server, and I'm going to be showing you both of these. Okay. So the first option is you have a desktop system, you have all of your files there. They're laid out the way that Hang Suda showed in the previous presentation. So you have the directory structure that has the study names and the experiment names and the file names. And then you want to actually run this. So the first thing you have to do just the first time around is download and install the client tools, and then do the initial setup where you tell it, what servers you want to use. So if you are going to use the desktop version, you want to install a tool called Deriva Upload. Deriva is the name of our software stack, and upload means it's uploading files. And there's a link here to a page that just has all of the download links for all of the different operating systems. So I'm just going to launch Deriva Upload. So the first time that you launch it, it won't know what server you want to use. So this configuration screen will pop up all by itself, and you'll need to set the server. You can also, just by clicking options, show this configuration screen. And I am, for the purpose of this demo, doing staging.goodmap.org. You would always use www.goodmap.org for the real system. And then there's this other configuration file thing that you can actually just ignore because you're not going to be using that. So that the first time through, you need to set that. And then every time you use the system, the first thing you need to do is log in. And clicking that login button will pop up the same window that has the same kind of information that you need to specify when you log in to the regular GUDMAP site. So I'm going to do that. And then I'm logged in, and if you look very carefully at this window, it will say logged in. And if you can't see it, just believe me, that's what it says. I don't know if there's a way to make this display actually any larger. The next thing you need to do is choose a parent directory. So this can be, you basically just need to have a directory called Deriva that's a subdirectory of whatever directory that you choose. So I have some things that I set up for the tutorial, and I have a directory called test underneath that, and that has Deriva. So I'm going to select this directory or folder. And once I've selected that, the system or the client will go through and it will find everything that looks like it's a file that I might want to upload. And the status here says pending, and again, I apologize for the size. Because I haven't actually uploaded anything yet. So when I click upload, what it's going to do is go through and look at all of these files, and parse their names and look for the corresponding records in the server to see if it can figure out how to hook it up. So, for example, for this file, it's going to look for a study called mouse IRI trap RNA-seq. And then it's going to, since there's nothing more under it other than the file name, it's going to assume that this is a study analysis file. Going a little further down, it'll look for a study called mouse IRI trap RNA-seq. And then because this right before that says seq, it's going to decide that it's an RNA-seq study and not an scRNA-seq study. So to look for an experiment called D14-IRI bound, and then look for a replicate called Q-Y5CA. Again, I think most people probably won't use these replicate RIDs, so you'll use something that has the replicate numbers. And again, what this client tool is going to do is it's going to query the database and make sure that there is basically there's a place in the database to attach this file to that corresponds to the encoded file name. So when I click upload, this is going to work for all except 2 that I have deliberately misspelled. Okay, so you'll see that these are all complete, which means that either the files were uploaded or this could be a restart of a previous upload. The other thing that it will do is checksum the file itself and see whether the file is there or not. If the file is there, it's not going to re-upload it. It's just going to say, oh, okay, it's already there, and move on to the next. And that's actually very useful if you find that you have, one problem in a batch of files, or if you upload a batch of files and then you later decide to add one more, it won't do a lot of unnecessary work. So most of these are complete. There are these 2 that have runtime errors. And those are a little difficult to parse, but basically what this says is that it can't find anything in the database that is associated with it. And if you run into something like that, what you would do is go through and make sure that you have a study with this name, which you do, and that you have an experiment with this name, which in this case we don't because I've deliberately mistyped it. So that's basically it for the graphical version of the command line of the download utility. You can log out and you can exit, but does anyone have any questions before I move on to the command line version?
Laura, just so I get this point across from my own understanding, that this is best for really large files.
It is, and I should have said this went through blindingly fast because these are tiny files that I have created just for demonstration purposes. But yeah, these are for big sequencing files. Okay. But it's a matter of preference. Okay, so I'm going to exit this. Okay, so then the next thing to do is, and that's what you do if all of your files are on your local system, but sometimes they're not. If you have a server where all of the files have been generated or processed or whatever, you have 2 options. I mean, one is you could download all the files to your local system and then run the graphical interface, but the other is that you can run the command line interface. There is one thing that you have to run on your local system though, and that is the authentication agent, because you're going to need to pop up that window to authenticate to the system. So if we go back to the top of this page, if you need to use the command line interface, you do need to install one thing on your local system first, and that's called Deriva Auth, and all that does is pop up the authentication window and give you a token. So I will run Deriva Auth. Okay, so the first thing that you want to do is make sure that you are authenticating to the system that you want to authenticate to. I have www.goodmap.org set up as my default system, but that's not what I want to use today. So I will select staging.goodmap.org. And again, if you want to add a new server, you just, that didn't work. But if you want to add a new server and you're not in the process of logging in, you can click that add and add a new one. So I have popped up the staging. I've selected the staging one to log into again. I just follow the normal procedure. And I'm logged in. Okay, now over here on the server, on the remote server, I need to have had the Deriva command line tools installed. So you may need to, if it's not a server that you run yourself, you may need to work with whoever does run that server. Again, the links to getting and installing those tools are on that wiki page.
So Laura, I'm sorry to chime in here, but just so I understand, this is a good example where we would have, most of us would have our sequencing files with our bioinformatics core, for example. The large files, the basic analysis is going to be done there. And then this, what you're describing to us here is we're authenticating to our GUDMAP or RBK server. And then we're also need a way for the bioinformatics server, say, of authenticating so that we can connect these two directly instead of downloading locally to our computer and then uploading to the GUDMAP or RBK.
Right, exactly. And this is especially something that you would want to do if you have really large files, because the download and upload is just a ridiculous thing to have to do.
Okay, thank you.
So, again, this, so this program is called Deriva Upload CLI. You need to say catalog to, this is just something that it has to do with the way that we've configured our service, and it's just something that you will always need to type. You'll need to specify the token that you got after authenticating, or through authenticating. The way you do this, unfortunately, is you basically just need to show the token and cut and paste it. So I've pushed the button called Show Token, which pops up a smaller screen that will offer to let you see the token. If you click Show Details here, you'll see the actual token, which I'm going to cut and paste. Now, you may notice that I have just pasted a valid authentication token into a presentation that's being recorded and will be published on our website. Normally that would be a really bad idea, but I'm going to show you also how to log out and invalidate that token, so it will no longer actually work for anyone once this is published. Okay, so Deriva Upload CLI, catalog to is something that you just always have to type. Token is the token that you've just generated, and then you can say, and then a directory. And I've made a different directory. So this has fewer things in it than the one that I showed you for the graphical interface. This is a little chatty. And it has uploaded the file. Now, this says upload successful. If I do the same thing again immediately, you actually won't see the upload successful because it's not bothering to upload it because it can see that the file exists and has the same contents. If I change the contents of this file, and then rerun it, you'll again see that it says upload successful. Okay, so I'm going to change the contents of the file again. But now let's just say I'm done with all of the uploads that I want to do. So I'll close this window that has the token in it. I'll click log out. And you'll notice that that staging.goodmap.org tab went away. And then if I, well, okay, that's big. Then if I try to upload it again, as you can see, this will fail, and it will tell me authentication required because the authentication token that I used is no longer valid because I've logged out. So that is basically everything. Does anyone have any questions?
Go ahead.
Just to confirm, because of the matchmaking process between the file names and the information on the server, you've always got to set up the experiments first before uploading the data. And what would happen if you try to upload a folder without doing the experiments information online first?
Right. If you try to do that, then what will happen is, you know, in each case, one of the first things that the client tries to do is determine where in the database this file needs to be attached. So if the records don't exist in the database, you'll get an error like one of the ones that we saw for those two that failed in the GUI tool. It'll just basically indicate that the system has no idea what to do with these files, so it's skipping them.
Okay, thanks.
But then if you create the records and run it again, it should work. But it will not upload any files if it's not able to determine a destination record for them. So every file on the system is going to be attached to some record that's already in the system. So in the case of the sequencing files, it'll either be attached directly to a study for the study analysis files, or it'll be attached to an RNA sequencing replicate file or an scRNA-seq metadata record. If it can't determine which of those things to attach it to, it just won't upload it. Does that help?
Yes, thanks.
So now I kind of hijacked the screen since we are getting close to. So this is pretty much in the wiki page. We do have, like, you know, an example of how you're supposed to name or create your directory structure, and also provide some example. So this is kind of the slide that summarizes that, and then so gonna do a quick. So basically, as Laura mentioned, you have deliver, right, as your folder, and then you put either seq or scRNA-seq underneath that to indicate whether this is a bulk sequencing file or a single-cell sequencing files, right? And then underneath that, you use a study internal ID. That is one of the metadata that you have to provide when you create study, right? And then use experiment internal ID. And then for the files naming, you have two methods of doing that. Either use the replicate RID. When you generate it, you will have an RID, or you use the replicate numbers to indicate, you know, which replicate to attach it to. And then use the file extension, like I mentioned, right? You can also have your custom text, like metadata that you want to actually have in your file name as well, but it has to be after, you know, either the RID or the replicate number. Okay, the same thing for the single cell files. So this slide kind of like summarize it. Okay, so either use like something like this to indicate replicate biological replicate number 1, technical replicate number 1, and then all the files associated with them, or use the RID. So so that's kind of summarizes structure. So another good thing that I like to point out is that we now do have kind of a submission dashboard that we summarize every month. So you can actually search based on, like, you know, which consortium, which PI, which data type, and you can see the different status associated with that month. So for example, in this case, you can see that the Doug's lab has submitted one study, right? And they are the study is in the submitted state, and then there's one experiment and one single cell metrics that are in preparation state. So that's kind of a good thing to know, so that you can just go back and then see how many files has been uploaded. And when you click on each of these blue text, it will lead you directly to the search page of that data type with that filter turned on. So this is all the examples and the documentation links that we mentioned so far in our presentation. And of course, if you have any feedback or you need help, just send us email. You can also access that through the navigation bar as well. I have one more thing to show, and this is going to be quick. So we have been working with a few groups now that that are actually trying to submit their paper to a journal, and so a good way to basically create a citable sequencing collection and then we include that in your method section or as a citable data entry data object is basically to create a collection of your sequencing study. And so I'm showing here is an example of that. So you can use collection as a way to sort of describe the goal of this data set, and then the detail. In this case, it's actually you can use it to, for example, you have figures and you have images in our system. You can basically then say that these are the actual figures that used in this paper. And then you can see that associated with the collection, there's a few types of data sets. These can be expanded further, and we plan to do that. But you can see that there's sequencing study collection, right? So again, if you click Add, then it will basically then pop up. It will pop up this screen so that you can choose from your existing study and include in the publication. I'm sorry, in the collection. And when you create a collection, right, there's a field called required DOI. If you mark it as true, we will then generate the DOI for you, and that you can use in your as a reference in your journal. So pretty much that is it. Questions, suggestions?
Hi, can you hear me?
Yes.
So I have a question about uploading FASTQ files. You have the file extension R1 or R2.fastq.gzip. How strict is that that R1.fastq.gzip requirement?
Well, we really encourage people to do gzip because it does actually reduce the file space. The file
Yes. So I'm asking because for 10x RNA sequencing, 10x genomics-based single-cell RNA sequencing, if I pull the files with the name ending like that and try to run it through 10x Genomics Cell Ranger, it does not like those file names. It needs after R1 and R2 _001.fastq.gzip.
Okay, so who's asking this question?
Gervais from Doug's Lab.
Okay, so we maybe can take this offline. So
okay.
Right now, I think it's strictly expect r1.fastq.gz.
Yeah.
You know, otherwise it wouldn't know what is this file about. So so maybe we can discuss and then talk about, like, you know, what kind of thing we can do, or you know, like maybe we can actually give you some suggestions on how to do it on your end.
No, I can change the file names, but if I'm an outside user who's gone to my paper and seen this, I want to pull these files and rerun an analysis, I can't just pull the files and immediately run it. I have to pull the file and rename every file in this study that I want to run in order to do the analysis myself.
So Yeah, so can we discuss this offline and see whether we can actually agree on the pattern that
Sure.
10x generate? Like, so
Okay.
if it's actually a very fixed pattern, I think we might be able to be flexible
Okay,
on that.
sure,
And
absolutely, no problem.
yeah, except either. So if you don't mind, can you send us an email of the file format that you get out of the 10x?
Okay, will do.
Any other questions or suggestions?
Just to emphasize that if you're looking for that wiki with the documentation and all that, you can always find it from either goodmap.org or rebuildingakidney.org under the help menu, submitting data. And that will take you directly to the homepage of the wiki. And we have a little sidebar that points you to different documentation, et cetera, which I actually update a little bit more. But that's a good way to remember how to access that.
Just one question on the geo. So I think Hong Suda mentioned earlier about metadata being exported geo. So I just wonder if you just comment or clarify what the plans are for integration with geo, or if people have the need to upload to both places, what this system can do to make it easier.
Yeah, so I think the plan is basically for you to you're going to have to upload to both places, but hopefully once you upload everything to our site, when you click an export, we're working on an export feature that will generate you this like zip file that hopefully has everything that you need to submit to Geo. And basically formulate all your metadata that you supply in the form that Geo will take, without you having to do any more work. The only thing that I can foresee is that we only acknowledge PIs in our data submission process, and that is because of the security policy decided by the PIs. So if you want to add more authors into it, you might have to do that manually.
Okay, great.
Cool. So I'll be sending out, after this, I'll be sending out the slides immediately, but I'll also be posting a link to the video of this. I just need to do a little editing. And if you have any feedback or questions, we can always be reached at help at either goodmap.org or rebuildingakidney.org.
Right. And if you need any help, just feel free to talk to us anytime. And if you want to include your data in any publication journals, for example, we're working with Doug's lab right now to help them put together a collection, and then we'll guide you through the whole process of data citation and everything.
And by the way, one more thing about receiving this information, I've been sending it to our mailing list, and it looks like I think there's some new people. So a lot of you, I just got back from vacation, so I've been seeing some emails from some new people. So I'll be adding you to those lists. But if for some reason you're not getting this info, please email the help address, and we'll get you squared away.
I think we are done. Todd, do you have anything to add or anything else?
No, I think that you guys have covered it. I do, in support of the idea about Geo, what we're really trying to do is reduce the amount of work. Yes, it's 2 uploads, and that kind of stinks, but we would like to get it to where we at least spit back out a form that Geo will accept readily. Now, that remains to be seen how well that'll work, but hopefully it'll save a little bit of energy on that side.
For the non-technical users, you can do the checksums in the export. That'll be a massive leap.
Well, that's a good idea, actually. Yeah, that'd be easy.
We do have the checksum and all that. Yeah, certainly. You're talking about the MD5 checksum, right?
Yeah.
Yeah, we do have all that currently in the record associated with the files. I think unless we have any other questions, then we should wrap this up.
Okay, great. Well, thank you again, everybody, for joining us, and we'll be sending you some more info soon.